Hello and welcome to my PowerPoint 2.2 steady state exercise. What is steady state exercise? Steady state is when the body is working at a steady state. It means that lactic acid removal is occurring at the same place as lactic acid production. Steady state exercise is activity that achieves a balance between energy required by working muscles and the rate of oxygen and the delivery for aerobic ATP production. Once we have been performing continuous exercise for a period of around 20 minutes, our body reaches a steady state. An example of steady state exercise would be going for a run or jog for 30 to 40 minutes at a continual speed and steady rate. If it is not a steady rate, then it would change to anaerobic exercise or high-level anaerobic exercise. How steady state exercise affects each exercise element. Before exercise, there is a rise in heart rate. This is the initial anticipatory rise. This is due to the release of the hormone adrenaline. This stimulates the SA node, and through the parasympathetic nerve, heart rate increases. When exercise is sub-maximal, a steady heart rate will be sustained and then plateau throughout the intensity of exercise as oxygen supply meets its demands. During steady state exercise, the working muscles need a continual production of oxygen and nutrients to reach them to ensure that they can work efficiently. Therefore, throughout exercise, the participant's heart rate will rise momentarily, but as exercise continues, it will plateau and remain at a similar rate because the heart pumps at a steady rate to deliver the oxygenated blood. Once exercise is terminated, heart rate will gradually return to their resting heart rate. Stroke volume. Stroke volume is the amount of blood pumped by one of the ventricles in one contraction. When exercising, there will be an increase in the person's venous return. This creates an increased volume of blood and stretches the cardiac muscle to larger than normal. The stretch of the cardiac muscle that surrounds the heart means that the heart can contract much stronger and therefore pumping out more blood than it usually could in each contraction. This is an increase of stroke volume. This effect on the heart is also known as Starling's law. Venous return is the rate of blood flow back to the heart. This is also increased. An average stroke volume for adults is 70 to 80 millilitres, whereas the average athlete can have a stroke volume as high as 110 millilitres. This is because their cardiac muscle surrounding the heart is much stronger and thicker, and is therefore able to pump out more blood in each heartbeat. The better a person's venous return, which is the blood's ability to return to the heart and fill, increases the stretch in the cardiac muscle, and therefore increasing their stroke volume. Starling's law. Starling's law states that the stroke volume of the heart increases in response to an increase in the volume of blood in the ventricles. This stretches the ventricular wall, causing cardiac muscle to contract more forcefully. The cardiac muscle surrounding the heart enlarges bigger than normal so that the heart can contract much stronger and therefore pumping out more blood than it usually does in each contraction. This is an increase of stroke volume through the volume of blood in the ventricles. Cardiac output is the volume of blood pumped out of the heart in 1 minute. During steady state exercise, there is an increase in cardiac output, as the working muscles need more blood to deliver nutrients and oxygen. This keeps them working at a steady rate. The cardiac output delivers more blood and is therefore increased, but delivers the blood through fewer beats. If a heart rate beats 70 times a minute and 70 millilitres of blood is pumped out each time, then the typical cardiac output at rest for an adult is 4,900 millilitres. Mo Farah has a heart rate of 33 beats per minute, and a typical stroke volume of a trained athlete would be around 110 millilitres. So an estimate of Mo Farah's would be 3,603 millilitres per minute. Blood pressure. Blood pressure is the pressure of the blood against the walls of the arteries and results from two forces. Systolic pressure is pressure exerted in the arteries when the heart contracts, and diastolic pressure is pressure exerted in the arteries when the heart relaxes and fills with blood. When exercising, the blood vessels dilate to ensure enough blood is getting to the working muscles, which therefore reduces blood pressure. This is then counteracted by an increase of blood pressure caused by the increase in cardiac output. When exercising, the systolic pressure is raised, but there is only a slight change in diastolic pressure. Vascular shunt. During exercise, the cardiovascular system redistributes the blood so that more of it goes to the working muscles, and less of it goes to other body organs such as the digestive system. This redirection of blood flow is caused by a mechanism called the vascular shunt mechanism. During steady-state exercise, the body works through vasodilation and vasoconstriction to distribute blood to the parts of the body that need it the most. When exercising in a steady state, the working muscles need more blood to deliver oxygen and nutrients. The blood vessels surrounding the working muscles will dilate so they can receive more blood, vasodilation. And the blood vessels surrounding other organs that don't need as much blood will constrict to reduce blood flow, vasoconstriction. Thermoregulation. Thermoregulation is homeostasis throughout the body's core temperature. When exercising, the body loses a lot of heat. It's important that the body is able to remove this excess heat so that the core temperature of the body does not increase. Excess heat is lost through sweating and vasodilation. When the blood vessels nearest the skin dilate and release heat. This is why we go red after exercise. The sweat is also that was also released cools down the skin after it has been excreted. Breathing rate. Just before encountering steady-state exercise, there is a rise in breathing rate. This is the anticipatory rise. The participant might feel a slight increase in breath just before exercise. This is caused by the release of noradrenaline and adrenaline. This causes the stimulation of the SA node through the sympathetic nerve and increasing heart rate slightly, which also increases a slight rise in breathing rate. When taking part in steady-state exercise, the breathing rate will increase at the beginning, as the body needs to take on more oxygen to ensure the muscles are oxygenated through the blood. It will then plateau and remain constant throughout the exercise, as the working muscles are getting a steady rate of oxygen as they deal with the demands of exercise. The average breathing rate for an adult is between 12 to 20 breaths per minute. Anything under or over these numbers would be abnormal. Here is a graph to show breathing rate in steady-state exercise. Tidal volume is air ventilated per breath. Exercise increases minute ventilation, which is the volume of gas ventilated in 1 minute. An increase in tidal volume refers to the quantity of air that is inhaled and exhaled with every breath. This increases at the beginning of steady-state exercise, but plateaus and remains constant throughout steady-state exercise. Throughout the steady state, the tidal volume will momentarily rise, and then throughout will stay constant, as a steady state of oxygen is being inhaled and exhaled to provide nutrients to the working muscles. Steady state exercise means that the body is able to inhale and exhale at a controlled rate. The average tidal volume for an adult is around 500 millilitres. Mo Farah has a much better tidal volume. This is down to the way he trains and the methods that he uses. For example, one way in which he trains is altitude training. Altitude training can be used by travelling to places such as Ethiopia, which is an average altitude of 4,232 to 9,843 feet above sea level. At these sorts of altitudes, oxygen is much more scarce and trains the body to use oxygen more efficiently. Another way to train in altitude is altitude masks, which are placed over the mouth to produce a similar effect. The mask forces you to inhale and exhale much more forcefully. Some say these don't work, and that's why it's important to travel to different places which have higher altitudes to really improve your aerobic capacity and tidal volume. Once at these altitudes, Mo Farah will train by running distances where breathing is much more harder, and therefore the body is training itself to use oxygen much more efficiently. Oxygen dissociative curve. The oxygen hemoglobin dissociative curve explains the amount of oxygen saturated within the hemoglobin. Most oxygen is transported in the blood by the hemoglobin. The hemoglobin is a red protein responsible for transporting oxygen in the blood. Oxygen in the blood reacts with the hemoglobin to make oxyhemoglobin. The binding of oxygen to the hemoglobin is dependent on the partial pressure of oxygen. This is when oxygen is inhaled and diffused across the lungs and into the body. Oxygen combines with hemoglobin in oxygen-rich areas of the body, such as the lungs. Changes in blood carbon dioxide levels and hydrogen ion concentrate cause shifts in the oxygen dissociative curve. These shifts enhance oxygen release in tissues and increase oxygen uptake in the lungs. During exercise, the blood becomes more acidic because of the increased production of carbon dioxide that we inhale. Hemoglobin, fully loaded with O2, is called saturated or associated, and O2 unloading from the hemoglobin is called dissociation, hence why it's called the dissociation curve. The shifts are different levels of oxygen released in tissues and increase oxygen uptake in the lungs. The right shift will be at steady state exercise because there is an increase in temperature and acidity. This is because we are breathing in more carbon dioxide. A greater dissociation is required to increase the supply of O2 to the working muscles. Energy system responses. This system starts with a build-up of ADP, which causes phosphofructokinase and glycogen phosphorylase to break down glycogen into glucose, which is then converted into pyruvic acid with oxygen. The pyruvic acid acts as a bind with phosphate to make 2 more moles of phosphate or coenzyme A, detects levels of oxygen and pyruvic acid, and creates acetyl coenzyme A. This is where stage 2 begins, also called Krebs cycle. The acetyl-CoA binds with oxalatic acid and creates citric acid. This binding is ultimately the Krebs cycle, with the binding creating more oxalatic acid, 2 more phosphate moles, carbon dioxide, and hydrogen. Then, this energy system moves into its third stage, the electron transport chain. The hydrogen from the Krebs cycle binds together with NAD and FAD, which are both transport enzymes. They then turn into NADH and FADH. This process creates 4 moles of hydrogen ions, which bind with oxygen to make H2O and gives 30 moles of hydrogen electrons. These 34 moles of hydrogen ions and electrons give us extra ATP for ATP resynthesis. During steady-state exercise, the aerobic energy system provides ATP at a slower rate than the other two energy systems. It is responsible for producing the majority of our energy while our bodies are at rest or taking part in low-intensity exercise, such as jogging. The aerobic system lasts for 3 minutes to an hour. Mitochondria. Both the Krebs cycle and the electron transport chain take place in organelles called mitochondria. The majority of ATP is produced in these organelles, so they are very important for energy production. They are rod-shaped and have an inner and outer membrane. Krebs cycle. Pyruvate acid from aerobic glycolysis combines with coenzyme A to form acetyl-CoA. Acetyl-CoA enters the Krebs cycle, which combines and reacts with a number of different compounds to produce ATP, hydrogen and carbon dioxide. Electron transport chain. The hydrogen atoms produced from the Krebs cycle enters this chain. The hydrogen atoms are passed along a chain of electron carriers and eventually combine with oxygen to form ATP and water. Neuromuscular responses. Muscle spindles are proprioceptors found in skeletal muscles. They detect muscle stretch and initiate a reflex that resists the stretch. Before the reflex, the message of the muscle stretch is sent to the CNS so they can initiate a response. As more blood is pumped through the muscles and excess heat is generated through exercising, muscle tissues warm up. The warmer the muscle tissue becomes, the more pliable it is. This means the muscles are able to stretch further without tearing. As the muscle tissue warms up, the rate at which nervous impulses are sent and received is increased, as the heat increases the speed of transmission. Here is a picture of muscle spindles in a stretched, relaxed and contracted position. The Golgi tendon organ. The Golgi tendon organ is a proprioceptive sensory receptor organ that senses change in muscle tension. During steady-state exercise, the Golgi tendon continuously sends signals to the CNS about the muscle's tension. Every time the muscle is tense during steady state, the Golgi tendon organ will send its message to the central nerve system, so the muscle will be able to ascertain more motor units and therefore is able to move. Here is a picture of the Golgi tendon organ. What is a hypothesis? A hypothesis is an idea or proposed explanation that you wish to test. A null hypothesis states the exact opposite of what an investigator or experimenter predicts or expects. An alternative hypothesis is a statement that suggests or advises a potential result or an outcome that the investigator or researcher may expect. The directional hypothesis is a kind that explains the direction of the expected findings. My hypotheses on heart rate, cardiovascular system, breathing rate, and energy system. Heart rate. I think heart rate will increase when exercise begins, and then heart rate will stay the same over the period of exercise, once the heart can reach the demand of exercise. When exercise is terminated, heart rate will decrease and gradually return to their resting heart rate. Energy system. I think that during steady state exercise there will be a low amount of lactic acid throughout exercise, with a slight rise in the middle of exercise. I also think that O2 saturation will remain steady and decrease towards the end. Breathing rate. I think that just before exercise begins there will be a slight anticipatory rise as the body gets itself ready for exercise. Then when exercise begins, breathing rate increases, and then exercise continues. Into the steady state, breathing rate will plateau. My method. Prior to the main part of exercise, we had our participant take part in a light warm-up to warm up the muscles that would be used. Just after the light warm-up, we took his heart rate, blood pressure, breathing rate, and O2 saturation. Our participant then began his steady state exercise. This was working aerobically between 60 to 70% of his max heart rate. We made sure he was working in this training zone with continued motivation. At 2 minutes of exercise, we took the participant's heart rate and breathing rate. Then at every 2 minute interval, we again took his heart rate and breathing rate. At 4 minutes of exercise, we took his heart rate, breathing rate, O2 saturation, lactate, and rate of perceived exertion. Then at every 4 minute interval of steady state exercise, we took his O2 saturation. At every 6 minute interval, we took his RPE, and at every 8, his blood lactate output. At the end of the 20 minutes of exercise, we took all measurements, and at post 1 minute of exercise, we took heart rate, blood pressure, O2 saturation, and breathing rate. We had 3 participants take part in steady state exercise. Here is a graph that shows their heart rate. Here is a graph that shows their systolic blood pressure on the left and their diastolic blood pressure on the right. I have removed one participant due to not being able to record their results. Here is their O2 saturation. Here is their blood lactate data. Here is their rate of perceived exertion. Here is their breathing rate. The 3 clients and normative data. Whilst 2 of the participants had results similar to the normative data, we found the other client had decisively different results. This was due to an experimental method. The client was working too hard and the group did nothing to stop this and let it happen. This meant the result of steady state exercise was incorrect, as the heart rate, blood pressure, breathing rate, etc. was not in line with the normative data surrounding steady state. Using the other participants' data, I can see that Josh's heart rate should have increased at the beginning of exercise and then plateaued. It kept rising. This was down to him working too hard and in a larger percentage of his max heart rate. Analysis of heart rate. My hypothesis was that I thought heart rate would increase when exercise began, and that it would stay the same over the period of exercise, once the heart could reach the demand of exercise. When exercise was terminated, heart rate would decrease and gradually return to their resting heart rate. Prior to the main exercise, we had our participant take part in a light warm-up to warm up the muscles that would be used. Just after the light warm-up, we took his heart rate. At 2 minutes of exercise, we took the participant's heart rate again, and then at every 2-minute interval we took it again. The heart rate of the athlete started at an average of 79 beats per minute. This then rises in between 170 to 180 beats per minute. The heart rates then plateau. They are able to cope with the demands of exercise, and the beats per minute are controlled at around 165 to 170 beats per minute. At 20 minutes, the exercise is complete, and therefore the athlete's heart rate decreases at a steady rate. At post 1 minute of exercise, the average heart rate is 153. This shows a decrease from the previous data. At the end of 20 minutes of exercise, we took all measurements, and at post 1 minute of exercise, we took heart rate again. Some data that we can disregard is Josh Messer's pre-exercise heart rate. This is at 104 BPM, whereas the other two athletes were at 58 beats per minute and 75 beats per minute. Josh's heart rate does not correlate with my hypotheses or normative data. It is far too high. This suggests he was working too hard. During the steady state exercise, the participants' working muscles needed a continual production of oxygen and nutrients to reach them to ensure that they can work efficiently. This is why the heart rate stayed at a similar rate for the duration of until the termination of exercise. Analysis of energy systems. My hypothesis for the energy system was measured through the oximeter and blood lactate monitor. I thought that O2 saturation would remain steady throughout the exercise at around 95 to 99%, and blood lactate would remain steady at about 10 to 12%, and then peak toward the end of exercise and lactic acid would be at its highest. We took O2 saturation every 2 minutes with the oximeter, which is placed on the finger and switched on. The blood lactate measurer was measured every 6 minutes with the blood lactate kit. A small amount of blood is taken from the participant and dabbed into the blood lactate equipment to create a reading. At pre-exercise, the average of the participant's blood lactate was 11%, and at 2 minutes this was at 11.2%. Then, as exercise continued, blood lactate rose to an average of 16.25%. This was because Max was working too hard, therefore his lactate levels were too high, and obscured the average of the data. We can also see that this was the case from his RPE levels. They were too high and therefore meant he was trying too hard. At 20 minutes, when exercise was terminated and the oxygen was now diffusing into the blood, this caused the lactate of the participants to drop to 11.35%. Against normative data, the average blood lactate in the middle of steady state exercise was around 10 minutes, should be around 10 to 11%. This was at 16.25%, and so I can deduct that someone, later found out to be Max, was working too hard and producing more lactic acid because of it. Analysis of breathing rate. My hypothesis was that just before exercise begins there would be a slight anticipatory rise, as the body anticipates exercise. Then, when exercise begins, breathing rate increases. Then, as exercise continues into the steady state, breathing will plateau. Prior to the main part of exercise, we had our participant take part in a very light warm-up to warm up the muscles that would be used. Just after the light warm-up, we took his breathing rate and the other measurements. At 2 minutes of exercise, we took the participant's breathing rate. Then at every 2-minute interval, we took his breathing rate again. At the end of the 20 minutes of exercise, we took all measurements, and at post 1 minute of exercise, we took his breathing rate and at one last time. At 2 minutes of exercise, the average of the participant's breathing rate is 30 breaths per second for 30 seconds, so 60 per minute. At 8 minutes, the average of the participant's breathing rate is 35, and at 20 minutes of exercise, when exercise is terminated, the average breathing rate was 34. From normative data, I know that their breathing rate at the start of exercise will increase due to the slight intensity increase, and the working muscles requiring more oxygen, so therefore the body takes in more breaths in less time. As steady state exercise continues, normative data suggests that their breathing rate should plateau as the participant's body are able to cope with the demand of more oxygen, and their breathing rate will slowly drop back down to its original rate. The participants' results mostly correlate with normative data. However, there is an anomaly with regard to Max's breathing rate. Similar to his blood lactate and O2 saturation, his breathing rate is far too high from the beginning of exercise. This could be down to Max working outside of his aerobic zone and instead working in an anaerobic zone, which would cause him to work harder. When taking part in steady state exercise, the breathing rate will increase at the beginning, as the body needs to take on more oxygen to ensure the muscles are oxygenated through the blood. It will then plateau and remain at a constant throughout the exercise as the working muscles are getting a steady state of oxygen as they deal with the demands of exercise. Finally, my evaluation of my hypothesis. For the majority of my hypothesis, I was correct in what I said about the outcome. For example, I said, I think heart rate will increase when exercise begins. This did happen for all of the participants who took part in exercise. However, there were some anomalies along the way. For example, part of my hypothesis for the energy system was, I think that during steady state exercise there will be a low amount of lactic acid throughout exercise, with a slight increase in the middle of exercise. As seen in my analysis of the energy systems, Max's lactic acid and RPE rates were far too high against the normative data. This was because of the human experimental error. If the exercise was carried out correctly, then my hypothesis might have matched the results. In conclusion, my hypotheses did not completely match the result that I got from the steady state exercise. Parts of my hypotheses were null hypotheses as they gave completely different answers to what I suggested they would be, and the other parts of my hypothesis were correct and lined up with what was going to happen. This is called a directional hypothesis. Thank you very much for listening.